So for my microbiology video questions, this is what I have to answer. The question is ciprofloxacin, erythrocymine, and alcyoclover are used to treat microbial infections. Ciprofloxacin, which I'll call Cipro, inhibits DNA grace. Ethrio, which I'll call erythromycin, binds in front of the A site on the 50th S subunit of the ribosome. Aclover is a guanine analog. The first question is, what steps in protein synthesis are inhibited by each drug? Cipro, which will inhibits transcription, but it has no DNA replication. Ethro, which the erythromycin is the formation of the polypeptide chains. And acleoclover uses DNA polymers to inhibit synthesis of DNA and RNA. The next question is, what drug is more effective against bacteria and why? Erythromycin, because it inhibits protein synthesis in gram-positive in the gram-positive only area and not in the eukaryotes. The third question is, which drugs will have effects on the host cells and why? Acleover will have the most effect on the host cells because it targets the viruses, which uses host cells to replicate. It will then inhibit the DNA synthesis, which occurs in the host cells, and attach the virus. And final, it says, use the index of our book to identify the disease for which acleover is primarily used. Why is it more effective than erythromycin for treating this disease? The reason why acleover is used for primary treatment for herpes disease, which stops the production of DNA. But erythromycin stops the polypeptide synthesis, and it avoids the viruses to be prevalent, and that way viral DNA replication should be stopped.